Okay, we've got quite a few of you. It's good to see you all again, those of you that I can see. We have quite a few things I want to do today, so let's get started. First of all, there will be a quiz 3, on Wednesday, a week from today. And that quiz will cover chemistry. It will cover biomolecules, which we'll do today. And it will include DNA and RNA, which we'll cover on Monday. So those 3 topics. And I'll send you an announcement when I send the recording for this session. I'll repeat this announcement about the quiz. Second point is that I have assigned a worksheet, which should be available to you now. It's a worksheet on biomolecules, and it's due September 12, which is next Sunday. And we will then discuss it on Monday.
So basically the things that are on that worksheet is what's going to be on the test, the quiz, Yolanda?
Part of it.
Oh, okay.
The worksheet will be just on biomolecules, and biomolecules is one of the topics that will be on the quiz.
This is Alex Alos. What exactly will the test be covering? Like, what should we study or prepare for?
Study the slides and the recording, and that will cover most of it.
Like, when you say slides and recording, are we talking about just today's slides or all of the slides since we started?
No, just the slides for those topics. So today is one slide set, and it covers biomolecules. There's another slide set, and you'll find these all in your modules in your home page. Another slide set covers chemistry, and there will be another one covering DNA and RNA.
Okay, so today's and last week,
yeah, everything since the last quiz, since quiz 2, and that will be those 3 topics.
Test on Monday.
This is Wednesday. It'll be Wednesday, a week from today. So it'll be again 10 points, 15 minutes. Okay, anything else?
Professor, so the test is covered them week 3, right?
This Gabriella?
Mm-hmm.
Okay.
It's for the week 3?
It's module, no,
Module week 3.
following week.
Okay.
Next week, you said, Professor, for test 1. The following week after that.
Repeat that, please. I have to turn my volume up. I can barely hear you. Okay, repeat the question.
All right, I'll repeat myself. So you're saying the test won't be next week? It'll be the following week?
It'll be next Wednesday, a week from today.
Okay. Okay. Gotcha.
Professor?
So next Wednesday.
Yeah.
Wait, I keep hearing test or quiz. Is it a test or a quiz?
I use them interchangeably. Quiz is always a short exam. The tests, what I'm calling tests formally, are full class periods.
So when is an actual test?
Coming up soon. Let's see. don't have it. Oh, yeah, so I've tentatively got it scheduled for Monday, September 20.
Okay,
So you can write that down as don't attend class that day. You'll have all day to take the test, and it'll be crafted to occupy about a full class period.
right,
Test
thank
one
you.
will be on Monday.
The 20th of September.
Professor,
Yes.
this is Rasha.
Uh-huh.
For the test, do you have one attempt or two attempts?
One attempt,
One?
Rasha.
Is a test time too, Professor?
Yes, it'll be a full class period.
Oh, okay. Thank you.
Professor, this is Alicia Carroll.
Uh-huh.
I am wondering, this is kind of off topic, but if you could go over the formula for the atomic number with and the mass number, how we talked about it last time in class, and you kind of gave us a formula on the slides, but I was wondering if you could talk about that a little bit more.
Okay, yeah, we are going back to that slide set today. And
Okay.
so when we do, remind me to of what your question is, and we'll go back to those particular slides and talk about them.
So
Okay,
the
thank
quiz is
you.
gonna consist with those formulas?
Say that again?
So is the quiz gonna have the formulas as well?
Yeah, the quiz might have formulas on it.
Mm-mm.
I haven't composed the quiz yet, so I don't know.
Oh, Lord.
Professor, this is Fakriya.
Uh-huh, okay.
So we do have like a study guide, right, for the exam?
The study guide is the homework, the grid that you did.
Okay. So we just have to study the, just only the packet, right?
No, this is why I have a problem with study guides, because it channels your studying and your thinking. I'd like you to have an understanding of all the material, not just certain items that I point out, although sometimes I do point out something that I want you to know. And I might say this will be on the test or the quiz. But in general, I want you to study the material that I present. And if you're familiar with the body of the material, you'll do well on the exam.
Okay. So do you know how many questions? Like,
The quiz will be 10 or fewer. It'll be 10 points. The test, I don't know yet. It'll be roughly twice that many, probably 20 some questions, between 20 and 30. But again, I haven't done the test yet, so I don't know myself.
okay, thank you.
I also had a question, Professor.
Uh-huh,
We can take the quiz at any time on Wednesday, right?
that's right.
Okay, will we be going over some of the material during class on Wednesday?
No, the stuff that's covered in the quiz will have been covered by Monday.
Okay, I see. Thank you.
So the material that we do in class on Wednesday will be new material, not on the quiz.
Okay, I see. Thank you.
I have a question, Professor.
Uh-huh. Your name?
Yusuf Faraj.
Okay.
Will tests also be taken the same way quizzes are taken? So like do we just take them at home technically without
Yeah.
being in class? Okay.
Right, you'll have all day from midnight to midnight. To log in, you'll click on the test, which will be under quizzes in Canvas. Canvas calls everything a quiz, but it'll be labeled test 1. You click on that and it will open up just like the quiz does.
All right, thank you.
Speaking of quizzes, I would like to share my screen with you and go over the answers to quiz 2. You should be able to see quiz 2 on your screen. If you can't, let me know. So there were, I think, 7 questions here. And I go over the answers for a couple of reasons. One is to give you the answers so that you know for any that you might have missed what the correct answers are. But the other point is pay attention to where these questions come from, why I ask them. And I'll show you in every case here, the part of the slide set, the part of the lecture that each of these questions came from. And so that gives you a clue. If you study the material that I cover in class, you'll be familiar with 90% or more of anything that will be on a quiz or a test. So the way to prioritize your studying is focus on the material in the slides. And if you need to go over the recordings in order to solidify that, that's a smart thing to do as well. Okay, the first question on quiz 2 is indicate statements that are components of the cell theory. It's taken directly from this slide where I've made a red circle around cell theory. So the answers: cells can arise only from pre-existing cells. Cells are the basic units of life, and all living things are composed of cells. So anything else on this list, it may not be untrue, but it's not a component of the cell theory. Now this next question, a lot of you had trouble with this. And so I'm referring back to the slides that this question is taken directly from, and those slides are here. They're about microscopes and what you can see with microscopes. So this is a matching question. Match the specimen with the type of microscope best suited. And I want to emphasize, whenever you read a question, read every word. So a key here is the best suited microscope to observe it. So if you look at the list, a plant cell, you could see a plant cell with any of these microscopes, but the best suited one would be a compound light microscope. And you see a picture of plant cells under a light microscope here in this slide. Bacterial ribosomes, what's the key here? Well, bacteria. Bacteria are very small, so you know you'll need a powerful microscope. And ribosomes, where are they? Well, they're even smaller. They're inside a bacterial cell. And so you'll want an electron microscope. And because you want to see inside the cell, you'll want a transmission electron microscope. And here you see pictures of a plant cell, seeing the internal structure showed by transmission. Fruit fly antennae surface features. So you should focus here on the words surface features, because I emphasized when we talked about scanning electron microscopes that that's what they do. They show surfaces. And so the outside of a cell, the fine structure of the outside of a cell, or any other object, is best seen with a scanning electron microscope. And so we want surface features of fruit flies. So even fruit flies are very large. You could see them with your naked eye. You don't need a microscope to see a fruit fly. But to see the fine structures of their antennae, the best microscope to use would be a scanning EM. The next one, virus internal structure. Okay, there are two keys here. One, virus is very small. That means you're going to use an electron microscope. Internal means it's going to be transmission. You want to see inside the virus particle. And finally, living amoebae. The key word here is living, because only with a light microscope can you see living material. Anytime you look at something under an electron microscope, it's going to be dead. You will have killed it in order to be able to look at it. So living amoebae, the best suited microscope would be a light microscope. I think I gave credit, some of you said dissecting scope, and that would work. It wouldn't be the best suited because dissecting scopes aren't powerful enough to see an amoebal cell very well, but you could see it, and it would, you could see a living amoeba cell. So any questions about this? The types of microscopes best suited for different purposes. Okay, number 3, fill in the blanks. Genes in eukaryotic cells are found in an organelle called the nucleus, whereas in prokaryotic cells, genes are not found in an organelle, but rather in an area of the cell called the nucleoid. Many of you got this right. In some cases, spelling was a problem, and if I understood what you meant, I gave you credit for it. So check your answers, and if you had it right and I didn't recognize it, let me know. Now, this next question comes from this slide, and I even have a little note here, important, remember this. And I think I mentioned during the lecture, this will be on the quiz. Some of you believe me and got it right. So the first part of the question is, we want to use a Latin binomial, and the first time it's mentioned in a manuscript, how would it appear? Well, you'd spell out the two words, bacillus and subtilis. Bacillus, capitalized B, subtilis, small s. And that could either be italicized or underlined. And if it's underlined, you leave a space between the words. The next part of the question was, how would it be designated subsequently in the manuscript? Well, in all subsequent mentions, it would be the same, except that bacillus, the genus name, is not spelled out. It's represented by just the first letter, B, and it's the same, I should say, it can be either italicized or underlined, not both.
I have a question.
Yeah, your name?
Trinity Daniels.
Okay.
If we forgot to italicize or underline, did we get the question wrong?
You got it partially right.
Oh, okay.
So, yeah, if you just wrote bacillus subtilis without underlining or italicizing, you got that part of it half right. Next, I think everybody got this one right. Fungi have stomachs outside their bodies. That's true. And the next one, most people got right. All living things are classified into one of three domains: archaea, eukarya, and bacteria, taken directly from this slide. And finally, a lot of you got this one right. Exobiologists hypothesize that you need Goldilocks conditions. These include an atmosphere, and so this question was taken from this slide. An atmosphere listed here, a liquid magnetic core listed here, an acceptable size, so the mass has to be not too big, not too small, and what else? And the what else came from this slide, that there must be water and it must be liquid. And so that puts a big constraint, because the answer here is you'd either have to have the correct temperature or liquid water. So I gave credit for either of those answers. Some of you wrote out phrases describing what you thought the answer was. And if you described it correctly, I gave you credit, even though you didn't use one of those two words. So again, check your answer. If you think you should have gotten credit and you didn't, let me know. Any questions about quiz 2 before we move on? Again, I want to emphasize that what I've illustrated here is how, for this quiz, I took every question directly from the slide sets. So for your studying, keep that in mind.
Professor?
Yeah,
Yeah, Alo's here.
okay.
I had a quick question, which was, what exactly are we supposed to read from that, like, concept, like, of biology book? How do we know what we're supposed to exactly read from it?
Well, I've indicated the pages that correspond to the slide sets. There's not an exact correspondence, and the purpose of the text and the homework, the worksheets, is to give you context. So the things that I think are really important that you should know are in the slides. But to give you a wider or perhaps even deeper understanding, you can read the pages in the textbook and look at the homework sheets.
Thanks.
Okay. Okay, I want to go over the worksheet that you completed and turned in. Go over this quickly because most of you got almost everything right. What is the charge on a neutron? It's none. Where is it located in the nucleus? A lot of you didn't get this next one right. How many electrons are there in the second orbital? There are 8. Many of you put 2. 2 is in the first orbital, but in the second and third orbitals, there are 8 in each. Name 3 types of bonds. Most of you got this: ionic, covalent, and hydrogen. Symbol for nitrogen is N. In a covalent bond, electrons are shared. In a double covalent bond, there are 2 pairs of electrons. Then a number of you did not get the next one. What type of formula would C6H12O6 represent? That would be molecular. Then describe how an ionic bond is formed. We got a lot of different answers for this. And I was pretty broad in what I considered correct. So almost anything you said that had anything to do with the answer, I gave you full credit for. But in essence, one atom loses an electron and therefore that atom becomes positive. Another atom gains that electron and therefore it becomes negative. And these two charges attract each other. And that forms an ionic bond. The water molecule polar, yes, and explain why. Again, you had a lot of explanations. If you were right or close to right, I gave you credit. But in essence, in a water molecule, the electrons are skewed to one side. The four most common elements in living organisms, I think you all got that right. Symbol for hydrogen is H. O represents oxygen. How to know the number of protons, it's equal to the atomic number. The three atomic particles are protons, neutrons, electrons. And then the last two questions are essentially asking the same thing. And that is, if you look at one component of an atom, it will tell you the numbers of the other components in a neutral atom. So any questions about that before we go on? I think you all did very well on this bit of homework. Okay, if nothing else, I'm going to click this off. Now I'd like to go back to and finish the slide set that we started last week. And someone had a question about chemistry. Could you remind me of what your question is? Something about formulas?
Hi, yes, this is Alicia Carroll. I was just confused about the formulas, kind of what you're talking about. On the bottom of our page in the book, what page is it? 28. It talks a lot about the atomic number and the mass number, and then there's a formula that goes along with it, how we can find the protons and electrons. I was just wondering if you could go over that again, so we could, so I could understand it, maybe somebody else could understand it better.
Okay. So. So let's start with this one, talking about protons. If we go back, so what's the previous slide? Okay, well, let's start back with this one. The three particles in an atom, protons, neutrons, and electrons. And then if we look at protons, and we look at the case of cobalt, in a balanced atom, the number of protons equals the number of electrons equals the number of neutrons. The number of neutrons can vary for isotopes, but aside from that, protons equal electrons equal neutrons. So does that help with your question?
Yes, that does.
And
Oh.
how you get the number of protons? It's the atomic number.
Yes, that helps. Thank you.
Okay.
I have
And so-
a question.
Yeah. Your name?
Yeah, Anthony Imolo.
Okay.
If you had the number of neutrons, could you also do like mass minus the neutrons to get the protons? Is that another way you could do it if you already had it, or?
Yes.
Okay.
Yeah. And a variation of what you just asked is here on this slide.
Yeah.
So for example, for cobalt, its atomic weight is given on the table. It's 59. And you know the number of protons from the atomic number, it's 27. So you get the number of neutrons just by subtracting. So 59 minus 27 is 32. And this is interchangeable as you've indicated. You can start with any of these and make a subtraction and get the others.
Okay, got you. Yeah, I was a little confused on the worksheet that we did, because it almost said like how to get the protons twice, and I was like, am I supposed to figure out the equation for this? Like, what do I do?
Yeah, no, that worksheet asked the same question twice.
Yeah, got you.
Excuse me, I have a question too. I'm Wasan Rafa.
Okay.
Yeah, and for the protons and for neutrons, they are like, the neutrons are more like all the time, but protons and electrons, they stay the same, like the same number as the atomic number.
Yeah, it's only the neutrons that can vary.
Mm-hmm.
Your first approximation of the number of neutrons is that it's the same as the number of protons, but in an isotope, it's not the same.
Thank you.
So we're all clear on this question, because if there are no more questions about it, I'd like to go to the part of this slide set that I didn't finish the last time. We were talking about hydrogen bonds and water. So let's start with this one, and this is a review. You saw this before. What is a hydrogen bond? It's a bond between a hydrogen atom, in this case, the little blue ball, and another atom that's electronegative. So a hydrogen atom has a slight plus charge, and an electronegative atom has a slight minus charge. And so the electrostatic attraction between the two makes a hydrogen bond. It's a very weak bond, but it's essential for life. And we'll talk a lot more about hydrogen bonds later today, in fact. So water molecules are hydrogen bonded to each other and to a lot of other things. But one of the things that makes water a unique type of molecule on this planet is this ability to hydrogen bond to itself. So what you see here are 4 hydrogen bonds. Each of them is between a hydrogen and an oxygen. Always between a hydrogen and an oxygen. This is a weak bond. And if you have a lot of water molecules, you'll have a lot of hydrogen bonds. And then I think we covered this last time, the properties of water. Important things to say here. One is 75% of the Earth's surface is covered with water. Cells are mostly water. It's a universal solvent, and it has a high specific heat, which means that it can absorb a lot of heat without itself changing temperature. Now, this is something that I'd like you to remember. It's very important because, first of all, water is the only substance on Earth that is found naturally in all three forms: solid, liquid, and gas. And what determines those forms? Well, ice, it's determined by hydrogen bonding. In the case of ice, every water molecule in ice is hydrogen bonded to every other water molecule. In the case of liquid water, some water molecules are hydrogen bonded and others aren't. So water is a mixture of those that are and those that aren't. Then if you raise the temperature further, it becomes a gas, in which case all of the hydrogen bonds are broken. The water molecules are just floating around free. So is that clear? I want to emphasize the importance of this, which I'll say more about that in a minute. So properties of water, high specific heat. Here's something that's very important. The solid phase is less dense than the liquid phase. And it's probably the only substance for which this is true. In most cases, the solid phase is more dense. So if you put a lump of lead, solid lead, into a pool of liquid lead, the solid lead would sink to the bottom. In the case of ice, the opposite. There's a high heat of vaporization. It's important, for example, when we sweat. It's a good solvent and it has adhesion and cohesion properties. Specific heat is the amount of energy required to raise the temperature of 1 gram of a substance by 1 degree centigrade. That is specific heat. So raising the temperature of water takes a large amount of heat because most of the heat is used to break the hydrogen bonds that hold liquid water together. And an example of the ramification of that is that because the Earth is covered with so much water, it acts like a giant thermostat. So when the sun's energy strikes the Earth, mostly it strikes water. And what does the water do? It absorbs that heat without changing temperature. And it keeps the Earth at a relatively constant temperature all the time, day and night. So at night, when there is no sun, the water temperature still doesn't change much. Now this elaborates a point I just made, that because of hydrogen bonding, ice is completely hydrogen bonded among its water molecules, and it floats. It's less dense than water, where the hydrogen bonds are optional. Some molecules have them, others don't. Then they keep breaking and reforming. And so what is the effect of that on life on Earth? So floating ice serves as insulation between the air temperature and the water temperature below the ice. So deeper in a lake, the temperature continues to get higher and higher. The deeper you go in the lake, the higher the temperature. And this permits aquatic life to exist in winter. Now, if ice didn't float, if it sank instead, lakes and oceans would fill up with ice, leaving no opportunity in winters for aquatic life to occur. So in this picture, you can see that the air temperature here is below zero. It's cold in these mountains. The ice temperature is exactly zero. The water temperature is greater than zero. So as at the interface between the ice and the water, the temperature is zero. And then as you get deeper, the temperature goes up. And again, this is due to the fact that water hydrogen bonds. When you sweat, why does sweat help? Well, when you exercise your muscles, you generate heat. That heat is absorbed by the liquid, the water on your skin. And since that has a high specific heat, you feel cooler because the water is evaporating. But the temperature of the water itself doesn't change much. Okay, these points are just reiterating the points I've already made. But now I want to help you understand what happens when a substance dissolves.
Wait, Professor, I have a question. I know it's a little not related to.
Diego Lopez, okay.
Do we actually know the? I don't know if you said this already, but do we actually know the actual origin of water, where it comes from?
Water is probably relatively common in the universe. If you remember, we talked about the Big Bang, and at first, all the matter in the universe was hydrogen atoms, and then they, as the universe cooled, they fused into helium atoms, and then they continued as time went on. And at some point along the way, water molecules were formed. That's the best answer I can give. Anybody else have an opinion or a thought about where water in the universe came from? Okay, now let's think about how something dissolves. And we'll talk about sodium chloride. Remember, sodium chloride is formed by an ionic bond. And so what you see here at the top is an undissolved crystal of sodium chloride. So the big green balls are chloride ions, so they're negative. The little blue balls are sodium ions, they're positive, and they're bonded together ionically. And now if you pour these crystals into water, what happens? Well, the water molecules are represented here by little red balls, which are the oxygen, plus little white balls, which are the hydrogens, H2O. So what they start to do is hydrogen bond, excuse me, not hydrogen bond, they form ionic bonds. The water molecules form ionic bonds with the sodium ions and the chloride ions. So you can see that here's a green chloride ion, it's negative. So it forms an ionic bond with a water molecule with the positive side of the water molecule. And similarly, here's a little sodium ion, it's positive, it forms an ionic bond with a molecule, but now with the oxygen part of the water molecule. And so that goes on until as you move down, you see that ultimately each sodium ion and each chloride ion is completely ionically bonded to water molecules. And at that point, the sodium crystal is dissolved and becomes a solution.
Hey, Professor, I have a question.
Uh-huh.
Yeah, it's me, Christopher. I have what does that word mean? Hydrophilic? I think that's how you say it.
Hydrophilic. Uh-huh.
Yeah. What is the definition of that?
Well, literally it means water loving. So you could say that sodium chloride is hydrophilic. It loves water. And you can see why, because it forms ionic bonds with water.
Okay. Is it similar to hydrophobic or no?
Hydrophobic is the opposite.
The opposite. Okay.
So we'll talk later about polar molecules, polar organic molecules, which are hydrophobic. They hate water because water is polar and they're non-polar and they don't mix. We'll talk about that in the next slide set.
Okay, thank you.
Now, two words that you need to remember. I've circled them in red here. Cohesion and adhesion. These are properties of water that, again, are part of the unusual nature of water. So what is cohesion? It's the tendency of water molecules to stick to each other, and again, due to hydrogen bonding. Adhesion is the tendency of water molecules to stick to other surfaces, again, through hydrogen bonding. Now, this has real consequences in living forms. So, for example, why does water move up in plants? And the tendency of water to do that allows plants to grow tall, to grow up and defy gravity. They can grow against the force of gravity because of the properties of water. And this is illustrated here in these pictures. The roots of a plant take up water into tubes called xylem vessels, and adhesion causes the water to move up. So if you think, if you stick a straw in a soda and you look at where the top of the soda is, or you could say water, the edges are higher than in the center, in the liquid in the straw. And the reason the edges are higher is because of adhesion. The water molecules are adhering to the walls of the straw. In this case, to the walls of the xylem vessel. Now, if we move up the plant to the leaves, water moves into the leaves through smaller and smaller vesicles. And at some point, it leaves the leaves. Plants are constantly transpiring. They're losing water. So they have to have a constant input of water because they're constantly losing it. And how do they lose it? Through cohesion, through the tendency of water molecules to stick to each other. So as a water molecule leaves through this little opening called a stomate, so water molecule leaves, it's still stuck to the one behind it. And so it tends to draw that out. And so as this goes off as a gas, the water molecules keep coming, and each one draws another water molecule behind it. So the effect of cohesion is that plants can transpire, they can lose water constantly, at least throughout the course of a day. So I want to make sure you are clear on this. First of all, what these two terms mean and the effect that that has on a plant. So what are some of the consequences? Here you can see a water strider seems to be able to walk on water, and in fact can, because of the surface tension of the water, which is caused by cohesion. And you can see here very tall trees are possible, again, because of both adhesion and cohesion properties of water. Shapes of molecules, I won't dwell on this. Molecules, these are space-filling models, and it just shows that certain bonds are flexible so that the molecule can twist and take on different shapes. And the final point here is that we're really studying organic chemistry, which are the properties of compounds that contain carbon. And you remember, carbon has 4 valence electrons, which means that it's always looking for 4, either to gain 4 or lose 4 or share 4. And so carbon makes up the basis of all life forms. And the final point I want to make is really something that you run into, in fact, every day of your life, and probably you don't think too much about it. So I want to make sure that you understand what is pH and how do you encounter it every day. pH is the concentration of hydrogen ions in solution. The range of pHs is between 1 and 10 to the minus 14th gram equivalents of hydrogen per liter of liquid. So pH is an exponent of 10. So pH numbers are a range of exponents from 0 to 14. And in this picture here, you can see the lowest pH possible is 0, the highest possible is 14. And how are those numbers arrived at? Well, a pH of 0 is 10 to the 0 power, and that by definition is 1. So pH 0 is 1 gram equivalent of hydrogen per liter. So this is very high hydrogen ion concentration. It's very acid. So pH of 0 would be like hydrochloric acid. The other end of the spectrum, pH 14, is indicated as 10 to the minus 14th. It's a very small number, gram equivalents per liter. So this is a very low hydrogen ion concentration, and this would be a base like sodium hydroxide here, which has a pH of 14. Now, in between, we have things that most of which you're familiar with. Stomach acid, remarkably, has a pH between 1 and 2. It's extremely acid. Later in the course, we'll talk about why. Other things you're familiar with, like lemons, vinegar, very low pH, which means high acid concentration. Apples and tomatoes, bananas are less acid. Milk is getting close to neutral. Pure water is neutral. It's a pH 7. Blood is slightly alkaline, as is baking soda and broccoli. Soap, bleach is very basic, alkaline, and a drain cleaner is even more so. Drain cleaner is primarily sodium hydroxide, usually. So because pH is critical for life, I think you need to understand what pH is and how pH influences life forms. Okay, I think that's the end of the slides of this set. So I want to leave this set. You see biomolecules, I hope. If you don't, let me know. What are they? The nucleic acids, proteins, carbohydrates, and lipids. That's what we're going to talk about for the rest of the period today. But before doing that, I'd like to look backward for a bit and recap what we've done so far in this course. So first we looked at cell structure and function, and we considered sizes. We went all the way from atoms, molecules, cells, organisms, planets, and we even talked about galaxies, again, sizes and distances. Then we talked about classification of organisms, and this really comes down to the use of these Latin binomials, which are critical for our understanding of how each organism, each species is unique, and how these are sorted out nowadays by their ribosomal RNA sequences. And we talked about analogous structures and homologous structures and the difference between how they evolved. And now we've just finished up considering chemistry and how chemistry relates to life. Atomic structure, what causes molecules to form. It all has to do with bond formation and the characteristics of various different atoms, of various different elements, so that they can form molecules. So we've just considered that, and now we're going to focus in on biomolecules. These are molecules found only in living things. So we're going to, each biological molecule exists as both a polymer and a monomer. The monomers in chains make up the polymers. So the monomer of a nucleic acid is called a nucleotide, and these are found in DNA and RNA. Proteins are peptides, and they're made up of monomers known as amino acids. Polysaccharides are long chains of sugar molecules made up of monosaccharides. And lipids are made up of fatty acids. And I'll talk about the structures of all of these things in this slide set. So there are two categories of nucleic acid, DNA and RNA. DNA is where the genes are stored. Think of DNA as a library. And that's pretty much all that DNA really does. It just stores information. RNA helps to extract that information and make it into useful molecules that can do work. So here's something that you need to know from top to bottom, the structure of a nucleotide. So DNA and RNA molecules are built from nucleotides, and each one has three components: a phosphate group, a 5-carbon sugar, and a nitrogenous base. So let's look at this picture of them. Where is the phosphate group? It's the yellow ball, PO4, phosphate. What's the 5-carbon sugar? It's the blue structure. Has an oxygen in the ring and 5 carbons. And the nitrogenous base can be one of 4. So in the case of DNA, this is a strand of DNA. The bases are thymine, here's its structure, adenine, cytosine, and guanine. And all of them together, the base, the sugar, and the phosphate, those three things are a nucleotide. And what either DNA or RNA is a chain of these. You can see 4 members of a chain here. And these chains can be exceedingly long, but they're always the same thing. DNA is considered a very boring molecule, or was for a long time, because it only had the variation in these nitrogenous bases, and then there are only 4 of them. Okay, DNA is a double helix. It's composed of 2 helical chains wrapped around a central axis. And here at the bottom is something that you must remember, that the A always bonds to T, and these are hydrogen bonds. And G always bonds to C. There's never any variation. G will never bond to T, A will never bond to C. And we'll talk more about the hydrogen bonds later. They're illustrated here by these bars. You can see the strands are the ribbons, and then there are bars between them where A is always bonded to T, G is always bonded to C, and the bonds here are always hydrogen bonds. Very weak, but essential for the structure. So what is the difference between DNA and RNA? On the left, you have an RNA nucleotide. Remember, I said a nucleotide has 3 components. And on the right is comparing that to a DNA nucleotide. The difference is in the sugar. DNA, or I should say RNA, has a hydroxyl at carbon number 2, and DNA does not have, it has just a hydrogen atom at carbon number 2. So the difference in the sugar is that RNA always has an OH, a hydroxyl. DNA always has just a hydrogen atom. And then of course, they differ by any one of the 4 bases. RNA is always a single strand. DNA is always double-stranded. So here are listed here on the left are some of the differences along with a similarity. DNA is always double-stranded, RNA is always single-stranded. DNA contains the base thymine. It does not contain uracil, whereas RNA contains uracil, but not thymine. And this is something that you'll be dealing with in your worksheet later. RNA molecules are short, where DNA molecules are long. DNA has hydrogen bonds, we talked about, shown here. RNA has no hydrogen bonds. It can't have because it's single-stranded. However, they both are held together by covalent bonds. So we'll switch from DNA and RNA to what is a protein. A protein is a polymer composed of monomers called amino acids. At the top here, you see 3 amino acids linked together, and the reaction that links them is called a dehydration reaction. I've got that here in red. You should remember that. Dehydration means that a molecule of water is lost in the formation of the bonds that hold peptides together to produce a polypeptide chain, which is in fact a protein. So to emphasize the point here about dehydration synthesis, these stick models show two molecules with hydroxyls adjacent to each other. H2O comes out, a water molecule comes out, and one oxygen is left, which joins the two molecules. So that is dehydration synthesis, and that's what forms peptide bonds. The opposite of that is hydrolysis. So if you start with two molecules that are joined together through an oxygen, and you add water to that, what you end up with are the same two molecules now with hydroxyls instead of being joined together by an oxygen. So this you'll come back to when you do your homework, and you'll have to draw the structure of an amino acid. And this is essential for you to know. Each amino acid has two components in common: an amino group, NH2, and a carboxyl group, C double bond O, OH. All amino acids have those two groups. Then what distinguishes amino acids from each other is the so-called side chain. So the side chain can be anything from a hydrogen atom to a complex structure like you see here in tyrosine or glutamine.
This would be the structure that we'd have to draw.
Yes, you'll have to draw this in your homework.
Okay. All right. Thank you.
Okay, now another thing you must remember is peptide bond and how a peptide bond is formed. So in the upper left here, you see two amino acids with the carboxyl end of one of them adjacent to the amino end of the other. And what you see is a hydroxyl and a hydrogen, which undergo a dehydration synthesis. So a water molecule is lost, and you end up with a peptide bond. Now, the peptide bond itself is this bond between the C and the N. Now, not all bonds between C and N are peptide bonds. For example, here is a C bond N that is not a peptide bond. So why is this one a peptide bond? It's because it's the C and the N are between a C double bond O and an NH. And you can see that illustrated perhaps better at the bottom here, where you have again the same two amino acids. The peptide bond is this bond between the C and the N, but it's a peptide bond because the C is a carboxyl and the N is an NH. So the peptide bond is a function of the context. If it's in this context, it's a peptide bond. If it's not, it's just another bond.
Steve Stinson.
Uh-huh.
Could you go back to that slide 1 second?
Yeah. Let's see. Yeah.
So I see the two lines on each of the peptide bond figures. What does that mean that it's two? The two lines are two bonds to that carbon and oxygen atom?
Yeah. That's a double covalent bond.
Okay.
And between the N and the H, it's a double covalent bond. And the peptide bond itself is covalent.
Sean here, Sean Koch here.
Okay.
Anytime we see this configuration here, an O double C to an HN in anything, that is a peptide bond then.
Yes.
Okay. All right.
Professor, I have a question too. This is Trinity Daniels.
Okay,
You said back at the other slide, we would have to draw like the amino bond or whatever for like our homework
uh-huh.
or the amino acid. Wouldn't even know how to do that on my computer. Do you have any suggestions?
Some of you who are more savvy than me, anybody have suggestions for how you do that?
Yeah.
I do that. Okay, go ahead.
If you're on Windows, you could probably just use MS Paint because they have like a little line and you can put in a letter. It's what I did for the other drawing we had to do for the homework. And if you're on Mac, there's probably, I don't know what it is, but they probably have something that's extremely similar.
Okay.
Yeah,
so you're saying I should probably like actually draw it somehow.
Oh, so you can actually draw it, and then I don't know if you have an iPhone, but I found out if you go into your notes on iPhone, you can actually scan papers and then like email them to yourself. So that also works really well.
Oh, thank you, Anthony.
Professor,
You're welcome.
Uh-huh.
this is Priscilla. I was gonna ask if like I can draw it on paper and like take a picture of it and like paste it into the worksheet. Would that work?
Yeah, yeah.
That works.
could do that. Yeah.
That's a good idea too.
To at least let you know we know what you're talking about, or that we're on the right track. So we can't actually draw it, but we can give you a picture of what we know it is.
Yeah.
Okay.
We are out of time, so I'm not going to be able to finish this slide set. Finish it on Monday and then go on to DNA and RNA. There will not be anything on the quiz on Wednesday that is not covered on Monday. So I doubt if I'll get through all of DNA and RNA on Monday, but any questions on the quiz will be covered on Monday.
Okay. Thank you, Professor.
Okay, thank you. And we are out of time. So good to see you all again. And
Thank you.
see you again.
Thank you, Professor.
Yeah, bye.
Bye.
Monday. Bye. Bye. See you guys Monday. Have a good weekend.
Okay, you too.
Bye.
Bye. Have a great day.
Okay, bye. Mm-hmm.